All right, hello everybody. So last class for Biology 107, just the final exam to do. So we're gonna do a little bit of review today. I've had some feedback from a few of you about some things you'd like to cover. If there's anything else that you're interested in, please leave some comments in the chat function and I'll see if I can get to them. Otherwise, I'm just gonna go through a few things and hope it's helpful for you, but like I said, I like to keep these things relatively flexible in case people feel they really need to focus on certain parts of the course. But otherwise, let's just get into this and we can do a little review. So first note to make is that I will have some office hours next week. The exam is Tuesday afternoon, so Monday and Tuesday morning, I'll have an open office hour. I'll have a Zoom link that I'll post on Moodle, so you can check that if you want to just drop in. If not, send me an email or make an appointment. I'll also be available over the next couple of days if you want to make an appointment. So one thing that's going to be different from the final exam that I forgot to mention the other day is that you will need to show me your photo ID. This is important that you are who you say you are for taking your exam. So hopefully everybody has a Keanu ID. I want something with a photo on it. If you don't have a Keanu ID, your driver's license is okay, but I would prefer if you use your driver's license, maybe you put a little piece of tape over any personal information like your address, because I don't need to know where you live. As long as it has a photo and your name on there, that is what the information I need to know. Don't need to know your height or weight or anything like that, or your driver's license number. So no personal information, please. Keanu ID is preferable. So when I do a sample lockdown browser test thing, it's going to ask you for your photo ID, so you can try that out at some point once I get that up. Okay, so I think I talked about this before. Nothing new here, we've all used this before. So same protocol, except for you're going to have to show me your ID. So these are the topics that are going to be focused on the final exam. And so I want to spend a little bit of time on some of these some of the core concepts and some of the things that people had asked for some clarification on. So we're going to get right into this. I'd like to say that one thing you can do is take a look at those sample questions and the study questions, and hopefully that will help and give you an idea to where to focus on studying, and also take a look at the study guide. Someone's asking if there's an update on the lab exam. I have posted the grades. As I mentioned, I might make some adjustments eventually, but it probably won't be till all the final grades are actually in. Partly because I'm just not going to have time to look at it until then. So let's get into things. I want to talk about DNA replication. And so I figured that was a good place to start. Of course, for DNA replication, we have 6 proteins and enzymes. And so I kind of just want to review some of the functions of those because it gets a little bit complicated. So I made this little table here, and you can see I have 3 of the enzymes there. I have DNA polymerase twice for a reason. I'll show you why in a moment. And I have what's going on at the leading strand and what is going on at the lagging strand. So let's just review these. So sometimes, by the way, primase is sometimes called RNA primase. You'll see that in some textbooks. Our textbook just calls it primase. And I've actually seen in one textbook them calling it DNA primase, but it doesn't make sense. It adds RNA. So that's exactly what this is doing. So this is adding an RNA primer. So adds RNA primer. And that's exactly the same thing as going on at the lagging strand. Adds RNA primer. I guess I didn't need to write that twice. I could have just drawn an arrow or something like that. Okay, so what is the DNA polymerase doing So go back and take a look at your notes. I know I made a lot of notes about what DNA polymerase is doing. I'm going to very quickly summarize it here because there's just not enough room to write everything all over again. But this is involved in the synthesis, or you could call it elongation, of the new strand of DNA. Okay, so that's the same for the leading strand and the lagging strand. So I know I have a note there as well, and because there's a couple of functions that's going on, right? So remember that, of course, the RNA primer is made out of RNA, and so that's another function of the DNA polymerase. Is it's going to replace the RNA primer. So we'll write a note about that. So replacement of the RNA primer with DNA. I guess we could also put in that this also proofreads. And what I mean by proofreading is if it makes mistakes, it will go back and fix those mistakes. That's part of what DNA polymerase does, which is very important. So the last enzyme there, ligase, doesn't do anything, so not applicable, the leading strand, but at the lagging strand, it joins the Okazaki fragments. Joins Okazaki fragments. There we go. Okay, so like I said, I always have a question on DNA replication. I always have a question on transcription and translation. All three of those things are kind of the major concepts for the last part of the course. Sometimes they're written, sometimes they're matching, something like that. I can't even remember what I did last year for replication. It may have been a matching type question where I had, something about the name of the enzyme or protein and then a function on the other end, okay? So make sure you know what these things do. 6 proteins and enzymes involved in DNA replication. So this is 3, right? 1, 2, and 3. So what are the other 3? The other 3 are shown in here, but let me just point out those ones we were just talking about. So there is DNA polymerase there. So notice this is DNA polymerase alpha. They have different names in eukaryotes and prokaryotes, but that doesn't really matter. But it is making new DNA, so it's also found right here. And there it goes, and that's the leading strand. We also have the primase, which is right here. And the primase for the leading strand, well, it's kind of off screen, right? So it's making the little RNA primer right here. And the third enzyme is the ligase, which is, where is it? Oh, there it is, right here on the lagging strand. So joining 2 Okazaki fragments together. So what does this leave? This leaves helicase. So what does helicase do? Helicase basically breaks apart the hydrogen bonds in the parent strand. So you can think of this unwinding. So I've heard somebody call it an unwindase once, which is actually not a bad name for it. It should be dehelicase or something like that, but I didn't name it. So helicase it is. Second thing here are these single-stranded binding proteins. So right here. So what are they doing? They're holding the DNA in a single-stranded formation until the replication is able to go through that particular segment of the DNA. And then the last thing is the topoisomerase, which sometimes has another name, which is called gyrase. So that may have slipped into the notes somewhere. So basically what that does is it relieves the tension. Due to the unwinding, things can get tangled pretty easily, and so it kind of untangles things. So 6 proteins and enzymes. So make sure you know all of them, for sure, 100%. All right, let's move on to transcription. So one thing that you must know is the difference between replication, transcription, and translation. So that's a place to start, okay? So transcription is the second part of this sequence, and this is RNA synthesis. So this is making RNA from DNA. I thought I had, it should say DNA right here. I did add it to the slide. I don't know what happened. I must have added it and then deleted it by accident. So there's the DNA right there, and we're making RNA. So this is transcription. So remember that this is done by an enzyme, and that enzyme is called RNA polymerase. And RNA polymerase does not need all those accessory proteins that DNA polymerase needs. So a couple of things about transcription to remember. Remember there are 3 stages, okay? Oh, I just lost my labels of DNA. 3 stages: initiation, elongation, and termination. So you're going to have to know what goes on at all those stages. And I'm just going to maybe mention that very briefly here, what's going on. But first, I just kind of want to mention this whole thing about the strands. Remember, we talked about 2 strands here, right? So we have the sense strand. And so what is the sense strand? The sense strand is the same, is the one that kind of has the same sequence as the messenger RNA. I would say kind of, because remember that our T's, of course, are converted to U's, okay? But besides that, it's the same, okay? And this is also the strand that's going to have the start codon in it. So the start codon is ATG, start codon. And remember, in RNA, the start codon will be AUG. Now, they don't show that here. This is the picture from the textbook. Now, here's a little hint for you. In the images that I make, I always put the sense strand on the top. For whatever reason, they flipped it in the textbook, but that's okay. It doesn't matter whether it's on the top or the bottom. But I just like it on the top because I like to read 5' to 3' on the top. That's just a preference of mine. But you may see it somewhere else in your travels. In my classes, you'll see it on the top. So what's the other strand called? The other strand is called the antisense strand, or the template strand. So you can use either term. And so why is this called the template strand? Because this is actually what the RNA polymerase binds, right? The RNA polymerase actually comes along here and it says, okay, I see an A, so I'm going to put a U. And then it looks here, you can see C, I put a G. Wherever they put in a C, another C, you put a G, an A, you put a U, and so on. So that's actually what the enzyme binds to. And then it makes the complement of it. So we don't need to worry about codons at this point. That's the translation part, but you can see that they're labeled there on the diagram. So initiation, I'm going to talk about here for a moment. Remember that in initiation, the RNA, well, that should be an N, the RNA polymerase, I'm just going to use an abbreviation here. So this binds the promoter. Okay, I'll define promoter in a minute if you don't remember what it is. Elongation is where the RNA is synthesized, and termination is where RNA polymerase binds the terminator. Binds terminator. Okay, so I have some, on the next slide, I have some of these definitions, promoter, terminator, and so on. So there's a bunch of words from the transcription unit, and I'm going to define some of them now. So some of them a little briefer than others. So RNA polymerase, hopefully you know what RNA polymerase is. This is the enzyme that synthesizes RNA. Now I could say a lot more about it. It, of course, makes the RNA complementary to the template strand on the DNA. And so that's kind of important. And it only goes in the 5' to 3' direction, so that's important as well. So what is a promoter? If you don't remember what a promoter is, this is a DNA sequence. It's a DNA sequence. I'm just going to use an abbreviation for sequence. And this is found before the gene. So it's be found before the start codon. And this is where RNA polymerase binds. And I'm just going to use abbreviation there. So basically it's a signal that says, okay, the gene's over here. Why don't you bind right before the start codon and we're all good. Okay, so that's where transcription begins. I don't have terminator here on this list. The terminator is kind of the opposite of that. It's after the stop codon. It's where the RNA polymerase detaches. So what's a transcription factor? So transcription factors, some of them are proteins, some of them are RNA and things like that. So that's why they're called factors and not proteins. So these are required for eukaryote transcription. Required for eukaryote transcription. So in eukaryotes, these have to bind before the RNA polymerase can bind. So some of them are tissue specific, some of them are kind of general, some of them might happen during different developmental stages, and so on and so on. So the transcription initiation complex, this is basically transcription factors. So TF for transcription factors plus the RNA polymerase. So like I said, RNA polymerase can't actually begin until the transcription factors are there. So once that whole thing is complexed together, transcription occurs. So 5' cap and the 3' tail. So these are modifications of your RNA. And so you can write that down as a definition. The 5' cap is at the 5' end. It's a guanine. It's actually methylated guanine. The 3' tail is a bunch of A's, so poly A. I can't remember how many of them there are. I think it's like 15 to 100 or something like that. It varies in range. But what's important? What do these things do? So these things stabilize the RNA. So what do I mean by stabilize? Well, RNA is kind of a wimpy molecule. It breaks down very easily. So these will actually help to keep it from degrading. They also basically signal this for nuclear export. So signal for nuclear export. So remember those nuclear pores. If you want to get out a nuclear pore, you have to have a signal chemical that says this needs to go out. Because the cell needs to know what's going in and what is going out, or else things end up in the wrong locations. The cell wants to keep organized. And the third thing, which is mostly the 5' cap, it's involved in ribosome binding. So ribosomes like to have things to grab onto. So ribosome, that's just one ribosome binding. And like I said, this is mostly the 5' cap. So it just gives the ribosome a little something extra to grab onto, because there's different types of RNA, and so the ribosomes need to know that this is actually messenger RNA. All right, one other term here, splicing. Okay, so what was splicing? So splicing is where basically introns are cut out. Introns are cut out of the messenger RNA. Way too much I could say about splicing, because there's a lot that's not really known about splicing, why it's done and all that. And we kind of had some side conversations about that, but I'm not going to get into it now. I do have other things I want to cover today. Okay, so if any of those words are kind of fuzzy in your brain, make sure you go home and learn them. Look them up in your textbook glossary, see what it says in my notes about them, because they're all revolve around important concepts that are important for different aspects of transcription. Some aspects are found in prokaryotes, some are found in eukaryotes, of course. Moving on to translation. Okay, translation, we are moving on, and now we're making proteins. And of course, this is a true translation because we're going from one language to another. We're going from the language of nucleotides to the language of amino acids. So remember that this happens at the ribosome. And at the ribosome, we have 3 sites. We have the A site, A for new amino acid. We have the P site, where the peptide bond is going to be made, and the E site, which is the exit site. So again, we have 3 steps. So we have initiation, where all of these things kind of assemble together around the start codon. So you can see there's my start codon there, AUG. And we have our start transfer RNA. So this thing here is a transfer RNA. And we have our first amino acid, which is methionine. So if you're going to learn one codon, learn AUG because it's important. It codes for methionine and it's a start codon. Elongation is the process of how all of this occurs, and I'm not going to review it right now, okay? But I do want you to go home and learn it, what's going on during elongation in terms of the sequence of events. We have the codon recognition happening at the A site, we have the peptide bond being formed, we have everything kind of shifting over, that's called translocation, and all of those steps. Lastly, we have termination. So that's where the stop codon arrives, and we have a release factor detaching this whole thing. So be prepared to explain some aspect of one of these steps, okay? I'm not going to ask you to describe all of translation, but there's definitely going to be a question on it. It's going to be worth more than 1 mark. Like I said, replication, transcription, and translation are all going to have questions worth a few marks on them. And I just have to decide what I'm going to do for it, because it's a little bit of a harder one to get around in terms of asking you to explain something. So I haven't decided what the format's going to be on that particular question yet. So one question that I've been telling you about, it's going to be worth up to 10 marks. I'm going to have you analyze a sequence of DNA. So what I'm going to ask you to do right now is get out a piece of paper, and I want you to work on this for a few minutes, and I'm going to give you about somewhere around 3 to 5 minutes to kind of work on this right now, and then we'll take it up. I just want you to have a chance to take a look at this sequence and be able to you know, try to do a little bit of on your own, because it's very important that you try to do a little bit of it on your own before I take it up. So I'm just going to pause right now, and I'll give you a warning when there's 1 minute left. So I'm going to take a look at this in a minute, but let me just go over a couple of things right now, just in case you haven't you struggled with it a little bit. You can see that the first couple of questions here are asking you to identify the strands, so the sense strand and the anti-sense strand. So I mentioned before that I will always put the sense strand on the top. That will not always be the case in your travels. But the other thing to do when you look for the sense strand, so let me just label that, sense strand, is always look for the start codon, okay? So that's the start codon there. This here, ATG, is not the start codon because that's not actually ATG. This is actually GTA. Remember, we always read DNA from 5' to 3'. So that would make this the template strand. Somebody has a question. They're asking about the genetic code. Yes, I will provide the genetic code for you. I'm not expecting you to memorize 64 codons. So if you find the start codon, we can maybe find some of the other aspects of this. I'm going to, I'll give you a minute to do the RNA and protein part, but I wanted to mention that the promoter is going to be before the gene, okay? So promoter, and in this case, you can actually see TATA, which is a pretty common motif that you see in a lot of eukaryotic promoters. The terminator is going to be after the gene. So I'm just showing you where all the triplets are, and I can tell you that this here is the stop codon. So the terminator is going to be over here somewhere. So I'm going to give you another minute here to work on the RNA and the protein part. So let's take a look at this. So hopefully you've made it this far, and there's my messenger RNA. So all I've done is I've taken the sense strand. So remember, the top strand was my sense strand, and I've just copied it letter for letter, except for all my U's, or all my T's have been changed to U's. So you can see I have T, 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 A. So I've changed that to U, 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 A, and so on. I've gone all the way down to the end of the gene. So the transcription starts before the start codon. So it's important that you do copy out the whole sequence. Okay. So I know it's tedious, but you know, it's not as tedious as doing a whole gene, which would be hundreds and hundreds and hundreds of nucleotides. So that's the first part, copying out the messenger RNA. And we always read our RNA from 5' to 3', so you can see I've put those labels on there. The next part is figuring out the protein sequence. So in that case, you're going to need your handy dandy genetic code, which I will provide on the exam. And you can see what I've done here is I've underlined where the gene is. Now, you may not know what your stop codon is, but usually what I do is I go along and I underline everything in triplets, right, until I get to the very end. So in this case, I would just kind of go like that until I get to the end. And then what I'm going to do is look at the genetic code and basically find all those codons. So AUG right there is methionine. So you can use the 3-letter code that's found on the genetic code. ACG, so I got to find that one there, right there. So that's threonine. GAA, so right there, that's glutamic acid. GLU. UUA, where is that? Up here in the top left. So that's leucine. AUC, okay, where is that now? AUC is right here. So that's isoleucine, ILE. And then UAA right over here is my stop codon. So that's actually my peptide sequence. You're not required in this class, but you know, it doesn't hurt. Beginning of a peptide is called the N-terminal or N-terminus because there's a nitrogen there for an amino group. And the end of my protein is called the C-terminus because there's a carboxylic acid which has a carbon in it. And that's kind of the convention for proteins. We don't have a 5' or 3' end, but they have an N-terminus and a C-terminus. Okay, so hopefully you got that. That's about maybe 4-ish out of the 10 marks, somewhere around there. And there'll be some questions around a few other things. I haven't written the exam yet, but some of them will be mutation questions. So I have a slide here that goes over some mutations, shows you an idea what mutation questions might look like. So this is the same DNA sequence. I've actually only copied the sense strand. And so you have a question like this, and you'll have to describe what's going to happen here. Okay, so remember, always look for your start codon, because that's kind of something important here, as well as your stop codon. So it says here, what type of mutation is caused by deletion at position 1? So in that case, the answer is nothing. It's not in the gene. It's before the start codon. So it might affect the promoter somehow. You could put that. I'm not expecting that for this class, but it's not part of the gene. So what about substitution at position 2? So let's take a look. We have ACG. I just want to find that on the genetic code. ACG is right here on the genetic code, that box that I'm just drawing right now. So if I change the A to anything else, I get a CCG, so I'd get a proline, a UCG, a serine, or a GCG, an alanine. So I'd be going from a threonine to something else. So that's part of the question, right? You have to tell me you're going from a threonine. Maybe I would actually say the question might say, what if I change the A to a U, right? Or something like that. And then you tell me it's going from a threonine to a serine. And the terminology for this is called a missense mutation. So make sure you give me a complete detail, right? Don't just say it's a new amino acid. That's like not a full mark. Be specific. Missense mutation has a technical term. Tell me which amino acid is changing to what for the full mark. Substitution at position 3. So in this case, if you take a look, if I change my G to an A, C, or a U, it doesn't really matter. So in that case, we're going from a threonine to a threonine, and that has a term, and that is called a silent mutation, where we're not changing the amino acid at all. There's one more there. So it says a deletion at position 4. So deletion means we're just cutting this thing out, right? So a deletion is technically called a frame shift mutation. But if you take a look at what's going on here, we have our ATG, and then we have an ACG, and then a GAA. So all those are the same. But then what I've done is I've introduced now a new start codon. So in that case, it's a little more complex of an answer, but you'd be saying that the gene has a premature stop codon, so the protein would be truncated, meaning it's not completed. And so that would be the full answer on that. So keep in mind on the exam, this part of the question takes a little bit of time to think through it. I could be looking at the template strand as well. And but these are the kinds of questions you're looking at. Like I said, make sure you be complete when you're answering the question. Don't just say it's a new amino acid, because that's not really that informative now, is it? So topic 18 is on gene regulation. And so this involves a whole bunch of processes. And we kind of broke it into 2 parts. We talked about prokaryotes and eukaryotes. So for prokaryotes, we mostly talked about the basic unit of gene expression. And hopefully you at least remember this word. The word is operon for the basic unit of gene expression in bacteria. So what is an operon? It's where you're regulating several genes at once. So it could be 3 or 7 genes. It just depends on the operon. And so it includes a whole bunch of things. So obviously it includes the genes. Could be 3 of them, could be 5 of them. It's going to include the stuff before the genes, so promoters and operators. If you don't remember what an operator is, we'll come back to that in a minute. Remember, we just reviewed promoter a few minutes ago. And then after the genes is the terminator, where the RNA polymerase is going to come off. So I have the tryptophan synthesis operon here. And what I want to do is kind of just go through all these numbered parts and see if we can remember what they are. So number 1 is the promoter. So remember, the promoter is where the RNA polymerase binds. Number 2 is the operator. So I need to find a space to fill in what an operator is. I'll write it down here. So an operator is a regulatory sequence. Regulatory sequence, where we'll call it a regulatory protein may bind. So by the way, there will not be a major question on operons. probably a maximum of 2 marks, so maybe multiple choice or something like that, because I know we covered it very, very quickly. But it is important to kind of have a bit of an idea of what it is. If you go on in biology, you will talk about operons in genetics classes like crazy. They're very, very important. So then we have our genes. You can see in this case here, we've got, see here, 5 tryptophan synthesis genes, and they're being read by this enzyme here. So that enzyme there is the RNA polymerase. And the genes here, we've got the start codon here. So start codon is AUG, and then we've got the stop codon. And each gene has its own start and stop codons. And then, of course, they're going to be, this whole strip of messenger RNA is going to be read by some ribosomes, and it's going to make these box things here, and these box things are the proteins. So that's kind of the big part of the operon. Now go over to the left, and you can see there's a regulatory gene that is going to regulate the operon. And I'm not going to get into all that because, like I said, it's just it's a little too much. We have other things to cover. We didn't really cover it too much, but you can take a look at the notes. There's a big section on it in the textbook, and kind of give you the 30-second explanation is that this gene over here regulates this whole operon over here. So this little protein that's made as a repressor protein that can bind the operator and block the RNA polymerase from doing its job. And it's going to be responsive to tryptophan. So there's a little bit of feedback regulation going on here. All right, where are we with time? I think we're doing okay. Just keep in mind, if you have any particular questions, please feel free to put them in the chat feature. So in eukaryotes, way more complex because there's so many other things going on. And I kind of just touched on a bunch of things. So I have some of them sort of summarized here, things that we talked about in class. So a lot of the regulation in eukaryotes is happening at the DNA level, at the transcriptional level. So some of it has to do with how the DNA is packaged. So remember that we can have euchromatin or heterochromatin. So remember, euchromatin is kind of the, it's not packed and condensed, it's more loose. And when it's loose, it's accessible by the enzymes. So if you pack your DNA and make heterochromatin, those genes are not accessible. So that's the first step, is to have your DNA available. There is a bunch of regulation that happens at the transcriptional level involving transcription factors and a bunch of other things called repressors and silencers and enhancers. And I had decided that it was probably not worth it this semester to get into all that because it's very, very complex. And I just really want you to focus on the basics and know the basics really well. And if you can do that, you'll be in good shape for your second year. So after the RNA is made, we have the capping and tailing. We've already talked about that. The RNA itself will have to go out a nuclear pore into the cytoplasm and find a ribosome. There is some regulation at the ribosome. One thing that we did mention is binding of the 5' cap. Translation is going to occur, and sometimes proteins are processed after translation. They might become glycosylated. So glycosylation is one modification that happens after the protein is made. And some of these proteins have signal peptides. So I want to talk about signal peptides for a couple of minutes because it was something that I know is a little difficult to grasp the first time around. And so it's worth it to talk about that. So I'll show you the relevant image from the textbook. So here was that image from the textbook. Let's dissect it a little bit here. First thing it says here, proteins that need to go to the ER. So endoplasmic reticulum. Remember, there's 2 options for a protein when it's being made. It will be made in the cytoplasm or it will be made in the rough ER. So if it is a protein that needs to go to the rough ER, it has something called a signal peptide. So what is a signal peptide? So signal peptide is kind of like an extra few amino acids at the beginning of a gene. So right after the start codon, they have a few more codons in there, so about 20 amino acids. And so it's at the start of a gene. and it's a bunch of amino acids strung together, so it's called a peptide, but it's part of the protein. And so there's our protein, right? It's got a whole bunch of amino acids, and there's the first few. So I know it's not 20, but I just was cramming things in there, and that would be my signal peptide. So the gene is being translated at the ribosome, and the first few amino acids, if it is destined for ER, the first few amino acids are going to have a certain chemistry. And what chemistry is that? They're going to be hydrophobic, a lot of them. Not all of them. I think there's usually a lysine in there, but basically it's a signal, right? It's a chemical signal because they have a certain chemistry to them. So like I said, it's just a bunch of amino acids being made, part of the gene, but these ones have a purpose, and the purpose is to say, hey, bring me over to the ER. And that's what's going on here. And then you've got the rest of the protein, which is going to do whatever it does. Could be an enzyme, could be all sorts of things, a receptor or anything like that. So back to this little diagram. There's our ribosome. You can see it's got some messenger RNA. And the first few amino acids there are the signal peptide. So there it is right there. Okay, so the translation is going. It's not quite finished yet. But the signal is there, and that is that hydrophobic kind of patch, right? And there's something that recognizes that, and that thing is called an SRP. So the SRP stands for signal recognition particle. And so that's a complex, and that complex is made out of both RNA and protein. And so it binds the signal peptide. So that's why it's called the signal recognition particle, because it recognizes the signal, which is the signal peptide. And so this thing basically grabs onto it and brings it over to the rough ER, and that's its job. So one other note about the SRP is it has RNA in it, and this was RNA type number 6 that we talked about. So what is SRP RNA? It's a component of the signal recognition particle. That's all you need to know about SRP RNA. So back to the diagram, you can see that it's basically doing its job. It's bringing this over to the ribosome, and at the ribosome there's a receptor, and there's a little channel, and a bunch of other things are occurring. I'll show you the rest of the picture right now. Basically, translation continues, and the peptide itself is delivered inside the ER. The inside the ER is called the lumen. And then from there, it's going to probably get glycosylated, put into a vesicle, and then it's going to go to the Golgi. And then from the Golgi, it's going to go to wherever its final destination is going to be. So there's not really any room in here, but I'm going to put a little tiny note here. This is going to a vesicle. And then the vesicle goes to the Golgi. And then from there, it goes to wherever else it's destined to go to. So a few other things here. I do have a short video I will show you on the signal recognition particle if it plays. Worked for me earlier. Oh, there it is. Okay, I'm going to play this for you. It's like less than a minute, and it kind of explains the signal recognition particle and the signal peptide. So here goes.
Many proteins need to enter the ER for modification with sugars. This occurs at the same time that they are being synthesized by the ribosome. Translation begins with synthesis of a short signal peptide sequence. A signal recognition particle, a protein complex, binds to the signal peptide while translation continues. The SRP then binds to its receptor in the ER membrane, anchoring the ribosome. The ribosome binds its receptor, and the signal peptide meets the protein translocator. Translation proceeds, and the protein passes through the translocator. The signal peptidase cleaves the signal peptide, leaving the new protein molecule in the lumen of the endoplasmic reticulum.
So that is it. So there's tons of videos like anything on YouTube nowadays if you're looking for some more clarification, or look at your textbook. I thought it'd be worth it just for me to show you a different depiction of this event. So there are the 6 types of RNA. So let's quickly review them. We have messenger RNA that, of course, encodes for the amino acids. We have transfer RNA that will deliver the appropriate amino acid to the ribosome. And for translation, ribosomal RNA has structural and catalytic roles in ribosomes. Telomerase RNA provides a template for telomerase to make those telomers. So telomers are, of course, extensions at the end of chromosomes, such as ours, that are linear. We had small nuclear RNA, which is involved in spliceosomes, has structural and catalytic roles. And then we have our SRP RNA, which we just talked about now, which is part of the signal recognition particle. So know these for sure. There's going to be some sort of question on this. Like I said, probably 6 types of RNAs. I could easily figure out some way to make it worth 6 marks. So make sure you know these, matching or some sort of fill in the blank. I'm not sure yet. So a couple of more things to cover before we finish. I had a couple of requests to kind of discuss these gels, these paternity and forensics gels. So you can have one or the other, either paternity or forensics, probably a paternity one. The forensics ones are almost too easy. So remember, what you're looking for in these gels is you're looking to see that if you're trying to figure out who the father is, that 50% of the DNA bands should match the child. So in this gel, it has the mom on there, which is a good control. So if you look at the mom and the child, exactly half of the bands match the mom and the child. So this is kind of a unique one. Notice the mom only has 3 bands, everyone else has 4 bands. Well, sometimes people have 2 of the same allele. So maybe this last allele, this one down here, and she has 2 of the same copies. That's hard to tell. But looking for about 50%, right? And then we can compare with dad 1 and dad 2. So let's compare with dad 1, and half of the child's bands match dad 1, and none of the child's bands match dad 2. So who's the biological father? Dad 1 is. There we go. Check, check, X. So let's look at a more difficult problem. This is maybe a little more similar to what you might see on the exam. Maybe this one's a little hard on the hard end, but just so you have an idea of what the wording might look like, or let's call the worst case scenario, right? So we're gonna have to pick apart these one by one. It says, we've got 5 individuals. It says what's consistent, with this. So it says here, 1 is the mom of 2, 3, 4, and 5. So remember, the mom is going to share 50% of the band. So let's take a look. One, it looks like 1 shares bands with 3, and another one with 3. So that's good. 1 could be the mom of 3. What about mom number 1 and 2? Actually, it looks like number 1 and 2 share no bands. So yeah, we're going to put an X there. Number 2, it says 4 is not the father of any of the others. Okay, I'm just going to see if I can erase that. Maybe I can't. Thought I could with this pen. Okay, so 4. Let's see here. Okay, so 4, it looks like, has bands matching with 1. It has bands matching with 2. Bands matching with 3. But no bands matching with 4, or with 5, I mean. Okay, so 1 is definitely not the father of number 5, but could be the father of 1, 2, and 3. So I'm going to put a check there, and maybe we better check the other ones as well, because I just wrote this now, and I meant there to be 1 right answer, but I think there's actually 2 right answers, but we'll see. Number C, 1 and 3 are not related. Well, we've already determined that 1 and 3 share 50% of the DNA, so that one is not correct. D, 4 is the mom and 5 is the dad. So we've already determined that 4 actually seems to be related to 1, 2, and 3. Now what about 5? 5 is related to 2, half the bands. 5 is related to 3 and half the bands. 5 is related to 1 and half the bands. So this is actually the answer I meant to be correct. I didn't realize I messed up with B. One more, let's look. 5 is the parent of 4. 4 and 5 are clearly not related, no matching bands. So that's what the question is going to look like, something like that. Hopefully maybe not that complex, but you get an idea of what it might look like. You're going to have to answer a question like that, and it's going to be multiple choice. So we're almost at the end here. Topic 19, of course, is on recombinant DNA technology. This particular unit, I want you to learn a bunch of definitions. So here's a few here. We have transgenics. We didn't cover cloning this year. We didn't cover stem cells, but we did talk about farm animals, GMOs. We talked about CRISPR. And also no examples of what can be done with these technologies. Topic 20 was on viruses, and so know about the structure of viruses and kind of a little bit about how they do what they do. And lastly, you should know a little bit about SARS-CoV-2 and the different types of vaccines, because there will be some questions asked on that. So we're at 4:50. A couple of more notes. I'll keep you one more minute and then I'll let you go. Make sure you take a look at those study questions. You know, I was kind of trying to take a look at the course in general and think of some general things. And one thing I did do is make a list of cellular processes. So I'll definitely probably have a question on each of these in the multiple choice section as I go through the units, because these are some of the sort of key concepts covered over the semester. And another key concept is looking at you know, some of the differences between prokaryotes and eukaryotes, so particularly in the last few units. So I think this is my last slide. So just want to wish all of you the best of luck. So thank you for taking my course. If you see me around in the community or at the college somewhere, please say hi to me. Let me know you're in my class. I probably won't recognize most of you because we haven't really seen a lot of each other face to face. But I'd love for you to say hi to me and tell me what you liked about my class, or if you have something you didn't like, I'd like that. Some feedback might not be too bad too, but hopefully you have lots of luck on your exams. If you have any questions, please let me know, and make use of my office hours on Monday and Tuesday if you need that, some one-on-one time. And when you're done your exams, have a great holiday and get some rest, and best of luck in the new year. So take care, everyone, and looks like that's a wrap for Biology 107.